It's a great honor to be here in this session. Thank you very much, Professor Norberto, for the invitation. And actually, since I come back to my home country in 2008, and Professor Griff is here, and he helped me a lot to do that, I was thinking about what is neuroscience. And in 2008, I said, maybe I can save the date for the next one. And I'm happy to be here in this meeting, in this session, with some important researchers like Professor Anders Kerner, that in this very interesting review gives some main lines for our study regarding the neurobiological approach of depression, and how can you understand a little bit better about the link between depression and seizures. And in this Lancet review, Professor Kenneth emphasized as one of the main points the hyperactivity or HPA axis, and this is one of our main focus of study since we work in the King's College London and developing these follow-up studies here at the University of São Paulo. Actually, we have a great influence of the group of Professor Decloé and Professor Marin Yogos, and try to understand a little bit about this in this very nice review about stress, hippocampus, and epilepsy, and which could be the next point, the next question about these links. And again, HPA axis could be one of our answers to understand a little bit about that. And so my talk will focus on HPA axis and how this so limited and so important disease, I can say it's a disease, depression, could be a little bit better treated and understand it. As you can see in this very nice review from Sibylle and French, published this year, depression is a very chronic illness, and you need to try to stop this cycle to avoid relapse and to achieve the remission of these patients. I have a link with the Netherlands, Professor Yogos. I did a master in affective neuroscience in the Maastricht, and I have an honor to learn a little bit with Professor von Braak. And one of his review approach, how can stress cause depression? In Brazil, we have a very big influence from North American psychiatry, and one of the main figures, probably Professor Anders can know, is Bob Post. Robert Post from the NIH developed these important studies about kindling and about sensitization to affective disorders, where the main, the first episodes, you can have a stronger triggers and precipitant of the first episode. In the following of the illness, spontaneous episodes could be developed, and so this could be a kindling model that could be linked with affective disorders, depression, and why not say that I will talk a little bit about bipolar depression as well. And so chronic stress is really an important point to increase cognitive dysfunctions, change emotions and change feelings in chronic difficulties. And as Bruce McKean showed in this very nice review and in all of his studies about stress and adaptation, you can see that major life events depends on how environment stressors and trauma situations, and then is one of the points that I will try to go deeply is early life stress, early life trauma in child and adolescent, and how this stress could be perceived and change behaviors according to the vulnerability of each subject. And so you can have a physiological response that could be some kind of adaptation, or could be the development of allostatic load. And so you have been working in this model for some years, and you try to understand a little bit how depression could be understood as a kind of stress in this axis, the HPA axis, and how these receptors, mainly MR receptors at the hippocampus and GR receptors, could be understood in the linkage of the release of cortisol, and why people with depression, and why not say with epilepsy, could have these changes in these main receptors of the HPA axis. Another challenge, and I'm here working, following some studies of Professor Griff, serotonin in the Raphael nucleus will have a link with this HPA axis, hippocampus, with the two receptors, MR and GR, hypothalamic nucleus with GR receptors, and pituitary with GR receptors as well. And so you can have a link with another neurotransmitter that is locus coeruleus with the noradrenergic input that could increase this system. And so the challenge is try to integrate in a comprehensive way the neurotransmitters and this HPA axis. In this is a very nice painting from Monet, and then you can see here a relation between a mother and her child. But even in this so harmonic painting, you can have some bomb of more than 700 synapses that could change sensory, language, and cognitive function, mainly in the first years, first months of development. So if you have strong situations like early life trauma, it is another painting like Picasso can show a different situation. Here you can see probably strong nightmare for the mother and difficult situation for the child. And so this will change this development. And if you think about maltreatment, it could be really stronger in early life stages of development. Maltreatment, abuse, neglect could really change the situation and probably in vulnerability, people with genetic potentials, probably the environment, the stressor, the early life stress, the maltreatment could switch on these points. I work in the King's College, and actually I still doing some studies there, and I have a collaboration with Professor Avshalom Caspi that really, in a very nice studies, have shown the relation between maltreatment and some kinds of vulnerability. For example, here, the serotonin transporter. And so this study shows clearly that early life stress could really change these kinds of subtypes of depression, for example. And Charlie Nemiroff did a very nice study with Christine Hine showing that childhood trauma may lead to neurobiological unique mood disorders, quite different from people without any kind of trauma. We did some studies in the collaboration with the Brown University, our group in London, Carmine Parianti, and the group of Linda Carpenter, and the University of São Paulo, Professor Del Porto and Andrea Mello. That is in the same line that the studies that Christine Hine published in the Psychoneuroendocrinology, showing that vulnerability could be linked with some situations of trauma, with early life stress, to develop changes in the central nervous system phenotype. And if this trauma are really chronic, without social support, you can develop subtypes of depression that could be followed in a physiological response. I really like this paper, and so for the students, I think is a nice review from Sonia Lupin, Bruce McQueen, and Christine Hine. I don't know the third author, Gunnar. And they show in a stress across lifespan how amygdala could be impairment, and how the HPA axis could change the program in frontal cortex, hippocampus, and changes the levels of glucocorticoids, and so how it could differentiate in different phases of the development, postpartum, adolescence, and even in adulthood. I'm a clinical psychiatrist, and so my idea is try to show how you can see this in patients. And the people from Columbia University, the group from Jonathan Stewart, have shown that depression with atypical features, with hypersonia and craving for carbohydrates, will be more associated with trauma history. And in this paper that they published in the Journal of Clinical Psychiatry this year, they show that more than 50% of the patient with atypical depression will have life trauma. And melancholic and other depression without atypical features will have very lower history of lifetime trauma. And so the history of trauma appears in patients with atypical depressions. And these patients will report more traumatic experience even before or in the following of depression onset. And so I like to make a short summary, especially for the people that are arriving now. Stressors activate sympathetic and HPA axis system. Glucocorticoids can regulate stress response through two different receptors, mineralocorticoid receptors and glucocorticoid receptors, and both of them are in at least a two-way interaction. If you have a chronic stress, the physiological problems will be demonstrated in the neuroendocrine dysfunction. Of course, this will happen in people with vulnerability, and so the stress response will be linked with genetic pattern. A Brazilian psychiatrist, Flávio Kapczynski, developed a very nice model about comorbidity of metabolic problems. And so you can see here recurrent mood episodes could change damaging in oxidative stress. And you can see in neuroimaging structural changes, and then in a clinical approach, you can see these patients with depression, with treatment-resistant depression. These patients decrease coping, and as I mentioned, Bob Post's studies, they will have a kindling model, and then it will be increased if they have life stressors, and then the cycle continue with a recurrent mood episode. But if you can see this paper that we published with Professor Wagner Gattaz and Rodrigo Vieira, that is a friend that is working in the NIMH in US, we measure plasma cortisol in naive bipolar patients in their first episode. And you can see that patients with manic episode will have lower levels of cortisol, and different patterns regarding if they have euphoric features or irritable mood. In this study that was published in American Journal of Psychiatry, you can see if the patients have several manic episode, you can see a change mainly in the ventricular area. This is quite different from patients that you assess that you have just first or very beginning of this illness. And so the chronic stress of bipolar will change structural areas like ventricular in many areas of our nervous system. I like to emphasize that 90% of these patients will have manic psychotic episode. I was just talking a little bit about this model that Pace and Spencer developed with Professor Yogos. And I believe that this is quite interesting. Probably you are going further than this, but in a schematic way, you can say that the MR receptors will be quite activating lower stressor intensity and novel intensity environmental, and then in stronger stressor with higher levels of corticosteroids, you have the activation of GR receptors. And then you can have two different patterns of illness. I was talking with you about atypical depression. You have a hyperactivity of the HPA axis with lower MR activity, with lower concentration of glucocorticoid. And you have an opposite pattern of depression, melancholic depression, with higher activity of the HPA axis and higher levels of corticosteroids with increased activity of the glucocorticoid receptors. Most of the studies have been done to assess the HPA axis with the dexamethasone suppression test. We developed a new test that can assess HPA axis in depression, that is prednisolone suppression test. Prednisolone different from dexamethasone because it links to both receptors. Dexamethasone links just to GR receptors, and PRED, you can assess both receptors, have almost the same pharmacokinetic of cortisol regarding half-life and bind to the same proteins. And so you believe that this could be a more physiological assessment of the HPA axis than dexamethasone or even the CRH test. We just finished last year this review about how to assess HPA axis regarding the two receptors in patients with depression and early life stress. And most of the studies will assess with dexamethasone suppression test. And you can see here this focus on DEX-CRH that could be one of the most used way to assess the HPA axis. And you can see that you have conflicted results when you assess human or man regarding cortisol or ACTH. And then we believe that dexamethasone is quite different from cortisol, as you can see regarding selectivity of GR and MR receptors. And so that's the reason that you are proposing prednisolone, that you can see that have a closer selectivity to cortisol. And you're assessing the MR receptors with fludrocortisone as well. And this is the paper that you published last month in the Psychopharmacology, the role of mineral receptor function in treatment-resistant depression. You assess 24 subjects, patients and controls, and use prednisolone to assess both receptors. And you assess them with spironolactone, that is an antagonist of MR receptors, and use both PRED and spironolactone to try to antagonize the MR receptors. We can find that in healthy controls, you don't have this problem. You can see that spironolactone increase cortisol, as you can see here, in a very nice way. Antagonism of MR receptors increase cortisol. The prednisolone suppression test suppress the levels of cortisol in assessment from 9 to 5 a.m. You measure salivary cortisol. But when you include the pred and spironolactone, you can see that look like dexamethasone suppression test. This happens in controls, but in patients, you cannot see this combination. The spironolactone didn't increase the cortisol compared to placebo. And the combination of MR antagonist and mixed MR and GR had no effects on suppression of cortisol. And so you could say that you have a hypercortisolism in these patients and MR malfunction, such as a downregulation in these resistant depressive patients. You are almost finishing this paper, and probably you submit it soon, a collaboration with endocrine professors here, Margaret Castro and Ayrton Moreira and Professor Greff. You did an assessment of these patients in the very beginning, and then 60 days after, you see the follow-up. In these 45 unipolar depressive patients, we found a sample with 70% with early life stress. And so quite high comorbidity of early life stress, a very different outcome. You can see that in red, patients with early life stress have a worse outcome regarding depression in 2 different scales. And the main result about psychometric, I should say, that is these patients, when you assess from suicide ideation, the patients in the very beginning of partial inpatient unit treatment, they have almost the same suicide ideation, but 60 days after, the patients with depression without early life stress decrease almost 0 suicide ideations, and the patients with early life stress still have strong suicide ideation. And when you split the sample in responders and no responders, you can see almost 80% of the patients will have early life stress. And so I should say that high stress and depression mood are quite linked with suicide behavior. And this could be something that you should try to understand, even if they have more comorbidity with epilepsy as well. This is our last paper that have been accepted in the Frontiers of Psychiatry. We use 2 different assessments in patients with early life stress and depression. We use GR and MR agonist. We use dexamethasone as a GR agonist and fludrocortisone as a MR agonist. And so you can see a difference just in patients regarding the use of MR. And so I would like to emphasize that the patients here are in a different pattern. They have lower cortisol level. Most of them are with atypical depression and personality disorders as well. And we split the sample with patients with without early life stress. You can see only a difference with the GR agonist dexamethasone with controls. When you assess 70% of the sample with early life stress, both receptors can see a difference in comparing with placebo. And so I'd like to conclude saying that these studies demonstrate that early life stress could lead to change in the HPA axis and may develop depression in adults. These findings show quite interesting point regarding the imbalance of GR and MR in early life stress, leading to hyperactivity of the HPA axis in this sample of patients with atypical depression. You could say that depressive patients without early life stress or treatment responders with better outcome will suppress more effectively with a GR agonist. But our data suggests that MR function is fundamental in the neurobiology and treatment response of these patients with early life stress. And so just to summarize in one of my last slides, I should say that the normal development, you can have a normal levels of GR and MR in the hippocampus. And on the other hand, patients that were exposed to early life adversity, you have lower levels of GR NMR in the hippocampus. And when they receive a stronger situations of stress or chronic stress, like repeated depressive episode or epilepsy seizures, this could activate this cascade here, and then the binding of GRMR in the hippocampus will be changed. And here, in the patients with early life stress, our hypothesis is that you have a poor binding in these receptors. And so you can have a lack of inhibition, and on the other hand, inhibition that could be very increased in the negative feedback. Alan Young, that is the head of the affective disorder now at the Institute of Psychiatry, saying that the future of the therapeutic targets in mood disorders will be the GR receptors. I like to add that could be as well the MR receptors. And we published a case series in the Psychopharmacology, Journal of Psychopharmacology, 2009. This was online. That in bipolar patients, you have improvement of response in bipolar affective disorders. They decrease the stress reaction when you use a mineralocorticoid receptor antagonist like spironolactone. You know, spironolactone is a very cheap drug used for cardiovascular hypertension, for example, and this could really help some patients. It's a great emotion, Norberto, to be here, and mainly with these very important researchers, and I like to choose one of these emotions. I'm not sure which one of them. I like to, maybe the first line here. And I like to just remember some memories and just say thank you to my group here. You have another PhD student that is not here, Vinicius, and I'd like to thank Professor Greff that helped me to come back to Brazil. Professor Tony Clear, that I'm still working with him, our patients, and mainly to the group, and Roberto that is very generous with young artists like this guy here, and not so young assistant professor like myself. Thank you for your attention.